So one of the drivers of inflammation that remains after taking care of the LDL and blood pressure and some of the other traditional risk factors that we have very good tools to control these days is what we can call triglyceride-driven residual risk. Triglycerides, they're a biomarker for a class of lipoprotein particles that we can call triglyceride-rich lipoproteins. These triglyceride-rich lipoproteins are very pro-inflammatory, much more so than LDL itself. And the current therapies that we have don't really address the triglyceride risk. So we have different baskets of risk. We have the LDL, that's well in hand. We have hypertension. We have tools for that. We can't do much about age and sex, but there's a big gap area in medicine today, and that is dealing with this triglyceride residual risk. So fortunately, we've learned the results of the REDUCE-IT trial, which is really one of the first large-scale trials on top of statins to show that omega-3 fatty acids can improve cardiovascular outcomes in secondary prevention. And part of this benefit might be from a relatively modest reduction in triglycerides. But we have a very exciting time coming up, again with Dr. Ridker and Dr. Aruna Pradhan. We have begun a clinical trial with a particular entity that we call a selective PPAR alpha modulating agent. Now that's a mouthful, so we call it SPARM for short. These agents have a number of effects. They do lower the triglycerides quite a bit, but they also lower some of the fellow travelers with the triglycerides, such as apolipoprotein C3, which we believe is one of the instigators of inflammation that associates with the triglyceride-rich lipoproteins, and a variety of other benefits. And it turns out that the particular SPARM agent that we're using appears from our current data to be much better tolerated and much safer, and certainly much more potent than some of the traditional PPAR alpha agonists that have been used, such as fenofibrate and gemfibrozil and bezafibrate. And these agents are very difficult to combine safely with statins. And it turns out that the agent that we're testing in this prominent trial, which is enrolling individuals with high triglycerides, with diabetes, and with a low HDL. This agent, which we call pemafibrate at present, seems to be very good in terms of the entire profile. It has few drug-drug interactions, and so far looks like a very clean agent. So we're very excited because we've made inroads against LDL. We have many of the other traditional risk factors under good control with current therapies. We're learning how to tame inflammation. But wouldn't it be nice to go to the root of the cause of inflammation, one that's very important in our current population, particularly those with obesity, visceral adiposity, and diabetes, insulin resistance in that constellation, which is sweeping the globe. This triglyceride residual risk